Go. So the endochondral and intramembranous ossification of bones. So the endochondral ossification is the process by which growing cartilage is systematically replaced by bone to form the growing skeleton. So it involves the replacement of hyaline cartilage with bony tissue. So in this process, the future bones are first formed as hyaline cartilage models. So during the 3rd month after conception, the perichondrium that surrounds the hyaline cartilage becomes infiltrated with blood vessels and osteoblasts and changes into a periosteum. So the osteoblasts form a collar of compact bone around the diaphysis. At the same time, the cartilage in the center of the diaphysis begins to disintegrate. So osteoblasts penetrate the disintegrating cartilage and replace with spongy bone. So this forms a primary ossification center. So ossification continues from the center towards the end of the bones. So after spongy bone is formed in the diaphysis, osteoclasts break down the newly formed bone to open up the medullary cavity. So then the cartilage in the epiphysis continues to grow, so the developing bone increases in length. So next is the intramembranous ossification. So it involves the replacement of sheath-like connective tissue membranes with bony tissue. So bones are formed in this manner are called intramembranous bones. So they include certain flat bones of the skull and so some of the irregular bones. The future bones are first formed as connective tissue membranes. So the osteoblasts migrate to the membranes and deposit bony matrix around themselves. When the osteoblasts are surrounded by matrix, they are called osteocytes. So the process, let's move on to the process of neuromuscular junction. So neuromuscular junction is also called the myoneural junction. So it is a site of chemical communication between a nerve fiber and a muscle cell. So a nerve fiber divides into many terminal branches. So each terminal ends on a region of muscle fiber is called the end plate. So it is embedded in the end plate or thousands of receptors, so which are long protein molecules that form channels through the membrane. So upon stimulation by a nerve impulse, the terminal releases the chemical neurotransmitter acetylcholine from synaptic vesicles. So then the acetylcholine then binds to the receptors. The channels opens and sodium ions then flow into the end plate. So this initiates the end plate potential, the electrical event that leads to contraction of the muscle fiber. So now the process of a muscle response. So the skeletal muscles respond to a single electric shock of sufficient magnitude by rapid, intense contractions called phasic contractions. So muscle, like other excitable tissues, has a period following its action potential during which the membrane will not respond to stimulation regardless of the strength. So now the process of major events of muscle contraction. So muscle contraction occurs when the thin actin and thick myosin filaments slide past each other. So in this conformation, the cross bridge binds weakly to actin and detaches so rapidly that it can slip from actin site to actin site, offering very little resistance to stretch. So the action potential in a motor neuron triggers the release of 2 plus ions from the sarcoplasmic reticulum. So then the calcium ions bind to troponin and cause trypomyosin to move, exposing binding sites for the myosin heads. So then the actin filaments and myosin heads from a cross bridge that is broken by ATP. So the ATP hydrolysis causes the myosin to swivel and change orientation. So then the swiveled myosin heads bind to the actin filament before returning to the original conformation. So the repositioning of the myosin heads move the actin filaments towards the center of the sarcomere. So the sliding of actin along myosin therefore shortens the sarcomere, causing muscle contraction. So for the process of major events of muscle relaxation. So relaxation occurs when stimulation of the nerves stops. So calcium is then pumped back into the sarcoplasmic reticulum, breaking the link between actin and myosin. So actin and myosin return to their unbound state, causing the muscle to relax.